Thank you, good morning. So good morning. Let me go through the techniques that we use in the lab to in the genetic for the diagnosis of genetic disease, and also sort of overview on how things evolved during the years, especially focusing on in prenatal diagnosis. So prenatal diagnosis, we all know from the 70s, has been done, diagnosis of chromosomal abnormalities has been done by culturing fetal cells and studying the chromosomes under the microscope to look at the structure and number of chromosomes. So the kind of accuracy nowadays we know is very high, approaching 100% in both amniotic fluid and CVSs, and CVSs maybe a little bit less than amniotic fluid, just because of phenomena that you will hear more about in the course of these days, like mosaicism. The main drawback of chromosomal of cytogenetic analysis basically is well, first for the lab it requires highly skilled operators, but also requires cell culture, so a couple of weeks to be able to block the cells while they are dividing and having a look at the chromosome under the microscope. And also for especially nowadays, what is the main drawback here is the low resolution, which is limited by the resolution of the light microscopy. One of the first things to try to speed up the prenatal diagnosis, at least for common chromosomal abnormalities, was using fluorescent in situ hybridization fish. This is basically using small bits of chromosome-specific probes, which are fluorescently labeled. And well, these allow by hybridize to their corresponding specific region to increase the resolution, especially in deletion, because you can see if there is a spot corresponding to that specific locus or not. One advantage is, of course, also this works perfectly without culture on the nuclei, so instead of 2 weeks, can take 1 or 2 days. The problem here is, again, you still need skilled operators, is labor intensive, quite low throughput, can be nasty to do this if the sample is not very good, especially in prenatal sample, where can be limiting, and needs also a specific indication. So for fish, you need to know what you are looking for and use the corresponding chromosome-specific probe. So we've done some work several years ago in trying to improve at least the speed of these analyses and simplify it for their routine use in high throughput laboratory. And this was by using qPCR or chromosome specific microsatellite. So this is how you, for instance, can detect Down syndrome with trisomy triallelic and diallelic pattern with all the informative markers on chromosome 21. This assay has a big advantages, especially for Aneuploidies over fish, and in some cases also even on the karyotype, because you analyze multiple loci, so you can also detect partial imbalance. It is very important that this is genotyping polymorphic microsatellites, so you can also exclude the presence of maternal cell contamination, and it is actually very important to do this test nowadays, even before any other molecular test like a chromosomal microarray or even sequencing or mutation detection, to make sure that you actually deal with truly fetal DNA without any maternal contamination. Can also detect mosaicism, is a molecular test, so you can include single gene defects. It's totally objective in interpretation, is a computer software analysis, easy to automate, so allows high throughput and reduced cost, and actually this is almost the standard for rapid aneuploidy screening in Europe since 2001. we are going from the resolution of the fluorescent microscopy in chromosomes, which on a good preparation can be anywhere between 5 and 10 megabases. Of course, depend the deletion, for instance, if it's a chromosome bound, might be easier to be identified. But now we are using chromosomal microarray, which is in principle is like performing fish experiments with lots and lots of probes designed all around the genome, so to scan all the chromosomes. The main advantage of this is basically, especially in prenatal diagnosis, you don't really need a phenotype to interrogate, because this is a whole genome analysis, and identify both microscopic and submicroscopic chromosomal imbalance, and is totally objective. At least the detection is done by computer software, and then has to be interpreted by skilled operators, of course. And nowadays there are so many different platforms available in coverage and densities for different applications apart than prenatal diagnosis. What are the drawbacks? Well, this does not identify balance rearrangement. In the prenatal field, probably this is not a drawback at all. It is an advantage. May uncover unwanted information, such as adult onset condition in a prenatal setting, absence of heterozygosity, which may indicate consanguinity. It may identify variants of unclear clinical significance. You can get a copy number variant, a small microdeletion or duplication, which has not been described before, causing counseling dilemma in parental anxieties. How this work? Well, we extract DNA from both the patient and the reference DNA of the same sex. We label these with different dyes and then mix them together and hybridize on an array. The array is a solid support, like a microscope slide, with loads of probes which are designed in a specific region of all the chromosomes. So these DNAs hybridize simultaneously. They compete each other in a way that if you have an excess of sequence from your patient, you will have an excess, let's say your green, as in this case. It could be one more chromosome 21. If you have loss from your patients, you will have an excess of color from your reference sample. So red, as in this case, could be chromosome X monosomy, for instance. So these spots are scanned and pictured by a Rai scanner at very high resolution. And then the ratio between the colors is measured by dedicated softwares. And as you can see here, well, you can detect both deletions and duplication, as in this case, for instance, was an imbalance resulting from balance allocation from one of the parents. Because you basically interrogate the whole genome, this can also be used to identify chromosomal markers, like in this case, is this extra chromosome here, ring chromosome, difficult to identify under the microscope, but on the chromosomal microarray you will easily see that this was a mosaic for the short arm of chromosome 12, which is polycercilian syndrome. So we are going from the chromosomes looked under microscope, like these two chromosomes look on computer screens, which can be sort of zoomed in as much as we want, depending on the platform that we are going to use on that particular cases. Another array platform is SNP arrays, which basically interrogates hundreds of thousands of small polymorphism, single nucleotide polymorphism all across the genome, where they add, on top of being able to detect deletions and duplication, they also can detect, as in this case, in this example here, uniparental disomies, only isodisomies, as in this case for chromosome 7, you can see there is no positive signal in the heterozygous genotype here. So between the two, they are exactly the same for chromosome gain and losses, depending on the resolution of both platforms that you want to choose. Triploidy can be detected by the SNP array, not by the array CGH, but this is not an issue if you have to do microsatellite to exclude contamination from the mother. In a sample, you will also detect triploidy before an array, so there is not even need to do an array CGH for triploidy. Mosaicism might be slightly easier to be visualized on a SNP array. You cannot detect uniparental disomy with array CGH, but at least the isodisomy can be detected with the SNP array, and no absence of heterozygosity for the array CGH, which in a way maybe in a prenatal setting might also be an advantage, because you might not want to uncover consanguinity in a family if you are not doing this test specifically, or eventually not increasing also, try to reduce results of uncertain significance. So as mentioned, there are lots of array design commercially available nowadays. Basically, the difference for the probes, which today are mostly only oligos, and how many probes you have on the slides. So how many points you can have on your chromosome, and how deep in the resolution you want to go. And then also on the design, how these probes are spread across the genome. This can be spread like a sort of uniform, as here, we call this backbone, only focusing, targeting regions which are clearly related to a particular phenotype, or actually a mix of both, which are the ones which are most commonly used today. We call this a targeted array plus backbone, like for instance, the most common is the ISCA 60K. So depending on the density of the array, you might think the more is dense and the more the resolution, the more I can detect. But actually doesn't seem to be exactly the case, because well, these are some papers with large study with chromosomal microarray, the first large studies, and you can see how well they add, as you all said, of course, more information compared to karyotypes, and in fetus with abnormal ultrasound, all these studies, they are almost around 6% more chromosomal abnormality in fetus with normal karyotypes. But interesting, the first paper here was a targeted back array with low resolution, and this other study here was a higher resolution oligo microarray. Same detection rate, but big difference instead in the number of variants of uncertain significance. So you might also want to check carefully the platform that you're going to use. As the array result, the array report that comes from the lab basically can be an abnormal result. This means has been identified a CMV which is clearly straightforward related to a phenotype. The alteration are basically not found in the control and very well described in the literature. A benign result, so if a CMV is found, this is not to cause a normal phenotype. It's found in control population with the same frequency. There are very common CMVs depending on population, which are basically polymorphic variants. And then the variants of uncertain significance, which especially in a prenatal setting is something we want to try to avoid as much as possible. This means the variant is not related with a well-described abnormal phenotype, but for instance, it can be large enough to per se be sort of causing concern. Not reported in medical literature, or not found in available database, or maybe only one study with not too much, not enough evidence. It doesn't contain disease-causing genes. Well, in all these cases, additional testing or further evaluation of the family is advised and performed, especially in prenatal. These findings used to be revised over time as new studies are published, and actually we also have to say that variants of uncertain significance that we were coming across 10 years ago were much more than the one that we are coming across nowadays, because most are reclassified either benign or pathogenic, the more the knowledge and the evidence is found and published. So, in a way, we shouldn't be too much afraid to also increase the resolution furthermore. Like we can do nowadays by sequencing the whole genome, which is something that you will, even in prenatal, which is something you will hear more about tomorrow. Sequencing the whole genome, well, we all know that the first draft of the Human Genome Project was finished in the year 2000. Since then, there has been a big push by companies to try to produce more efficient chemistries and more efficient machines to increase the throughput and the efficiency of actually sequencing. And basically everything started in 2004 when Roche came out with a parallel pyro sequencing system which pushed up by six-fold the throughput of sequencing. Since then, there's been a sort of rush between companies, and especially from Solexa first and Illumina then, which bring us in the situation that we know nowadays, with a huge decrease in cost per sequencing, starting from the introduction on the NGS, which basically happened between 2007 and 2008. Even, as you can see, very well outpacing Moore's law, and starting from the $100 million that was theoretically costing a single genome at the end of the Human Genome projects, we already breached the barrier of only $1,000 in about 16 years or something. So this is something that is available also nowadays to almost basically every diagnostic laboratory. This slide is only just resuming a few of the most popular instrumentation. Illumina, as mentioned, is by far the most popular. And also, don't take it as this, because figures are evolving so rapidly that some of these probably are already out of date. I only want to show you that there are different NGS, next generation sequencing approaches based on the detection system, and they all have some strength or some drawback. So depending on what you need these machines for, you might want to use one or another. It's totally, as I told you, even without a new model from, let's say, Life Technology or Illumina, even without waiting for a new machine, 6 months after you might have a new chip or an update in chemistry or a new flow cells, and the same machine will be faster and with a higher throughput. So the common step in all these next generation sequencing approach, well, the first difference basically with Sanger sequencing is in Sanger you first have to do a sequencing reaction and then a detection, which is usually done by electrophoresis, capillary electrophoresis. So these are two separate steps, which of course compare with NGS, which is both are performed at the same time, so it's faster and can produce more data in the same time frame. Also, these detection and sequencing reaction, which are simultaneously, are also performed all together on a huge number of different templates, and this is why we call this massively parallel sequencing. We are also actually already in a third generation where we don't even need to synthesize DNA because the sequence is performed at the single DNA molecule level, for instance, by analyze how the electricity change when these flow through a small pore. Another common step, of course, you need to prepare your template for sequencing, and this is called to prepare a library. So the DNA needs to be fragmented. Platform-specific sequences are added to all your fragments of DNA, and this is basically to stick the DNA on a solid surface, so to allow this to be then amplified in an emulsion PCR, for instance. So the library is then amplified through PCR, and we know that we need this because every single DNA fragment that we want to sequence has to be clonally multiplied to be able then, when it's sequenced, to actually detect the signal at the same time. And this is actually probably the limitation of next generation sequencing, which is when you amplify all your molecules, you can desynchronize. So instead of having a bunch of exact clone of your original template, you might have a mix of different products which are with different extension, so they can cause background noise and sequencing errors. And of course, the longer is your template, the more likely is this to happen. And this is why NGS platform used to produce a shorter sequence rate compared with Sanger sequencing. Also, the amplification can introduce bias, so depending on what you want to include and sequence, you need to stick to different experimental designs, like using different PCR buffers for a CG-rich region, etc. So the fragment DNA has to be cut in short pieces, and then at both ends of the short pieces, we stick these, to prepare the library, we stick these sort of adapters. Adapters are oligos, synthetic oligos, used to then use this DNA on the specific platform. This can also include what we call barcodes, so indexes, and these are unique to each DNA sample, can be up to 96, for instance, and these are very helpful because then you can mix up different patients, different DNA, and sequence them all together, which is very useful as these machines are becoming so powerful and capable every day that you need to fill them in as much as possible to try to reduce costs. So this is how an Illumina flow cells work. I'm just showing this because it's by far the most common in routine laboratories. So in the flow cell there are small oligos, stick, and these are complementary to the one that we stock on our DNA template. So when we put the DNA on the flow cells, the adapter recognizes the small sequencing on the flow cell, so they hybridize and can be extended by a polymerase. Once this is extended, it gets stuck on the original oligo, which is on the flow cell. It binds, hybridizes to the adjacent oligos, which are complementary to the other end of your fragments. Again, is re-extended by a polymerase, and so we end up with both the forward strand and the reverse strand sticked on the surface of the flow cell. The thing is, of course, this happens simultaneously for hundreds of sequencing adjacent regions, and then the reverse strand is cut. You end up with only the forward strands, and you can basically proceed to the sequencing reaction. So the process that I showed you before is to generate the copies of your a single DNA molecule, and this is called cluster. The clusters are stuck, of course, on the flow cell, and they have now enough molecules here to produce a fluorescent signal during sequencing that actually can be spotted by a camera, can be read, excited by a laser, read by a camera, and take a picture. And this happened when so the sequencing primers extends with a nucleotide at one time for each cycle. So if a blue is detected, for instance, as in this example, this corresponds to a T. If there is a red signal, it corresponds to a C, and so on. So basically, every cycle, one nucleotide is added, a picture is taken, the nucleotide then is deblocked, so it can be extended again. Good thing here is that this can happen in millions of clusters simultaneously at the same time, which are imaged on the same with the same picture. So all these clusters then can be, as I showed you, can be read one after the other sequentially, and the fluorescent signal is actually outputted in a sequence. One more improvement of this is also sequencing. It's called parent sequencing, and this is sequencing all your DNA fragments by both extremities. This is actually very useful for basically for two reasons. First, if a random DNA fragment you sequence, let's say 36 base pair at the 3' and at the 5' ends, then once this is aligned against the genome, you also can see what's in the middle and exactly how long it was your fragment, so you can determine accurately the size of these fragments. But also, when you align sequences a pair, well, the alignment is much more efficient, so you can have much more sequencing counts, which are efficient, useful for depending on your application. And this parent sequencing approach is proved to be very helpful, because by doing this, first has been discovered that in maternal plasma, the fetal DNA, the DNA from the fetus is shorter than the DNA from the mother, and actually counting only short fragments can improve what is the NIPT, so the screening of aneuploidies, especially in cases where you have much lower fetal DNA, like trisomy 18, for instance. And this is the pixel. This is the size of DNA fragments, which are mostly of fetal origin and non-maternal. So in the diagnostic laboratory nowadays, we have, most laboratories have this platform available, and the tests which are most commonly used in diagnostic are, well, by far, I think the random sequencing of maternal plasma for screening of aneuploidy, which is a NIPT, is probably what nowadays is the most, in terms of volume, used NGS-based test. Targeted gene panels. This means targeting a specific set of genes from 20 to 90 different genes, which are used to be related to a defined clinical phenotype, like a panel for skeletal dysplasia in fetus with abnormal ultrasounds. And clinical exome, this comprehensive and targeted sequence of regions within the exome, which are very well known and characterized to be associated with a clinical phenotypes, or also the whole exome sequencing, which include basically all the entire exome, and whole genome sequencing, which is, as Yuval mentioned, of course, sequencing both intronic and exonics. So whatever platform you use, you end up with a sequence of DNA. Like in this example, it's clear that you can target the power of NGS when in very specific area. Like in this example here is the exons of a gene. You can see all all the sequencing counts and not in region that you don't want. And the more you go on the same regions, on the same nucleotides, this is called sequencing depth. And the more depth you need, depending on the application, if you are looking for rare variants, for instance, you need more sequencing depth. So you can, of course, identify sequencing variants. The problem is, now is very easy to sequence the whole exome in the lab. The human exome is only, in average, less than 150 base pair, and the whole exome accounts for about 50 million base pair. So with the power of equipments that I showed you we have in the lab, this is within the easy reach of almost all of these equipments at very reasonable cost. So it can be done very, very quickly. The problem is, if you're sequencing the whole exome, you have out of the box, as soon as the sequencing is finished, about 60,000 variants. Most of these are corrected on the same software, on the same platform. Then you end up with rare coding variants, about 1,000, which needs to be filtered to create what is usually a short list of about 50 per case, and then this one has to be manually curated one by one by expert technicians, which is actually what is nowadays causing probably the pipeline. So this is basically to arrive at the definition of only one or two variants. So basically this is nowadays the cost of the interpretation is by far more extensive than the cost of sequencing, which is only just starting of just the start and the first part of this kind of analysis. So this is probably, as you all know, creating a whole new economy, which is how we handle all this data that we can generate very, very fast. And some application will be discussed in detail in the course of this meeting. Thank you.